not too much of a guest, but she can come on up here. Our speaker is Bethany Campbell. And Bethany is a 2018 King graduate. She majored in biology. And she is currently working on her PhD at UT Knoxville in biochemistry, cellular and molecular biology is the name of the program. So she's going to talk about how she decided to go to graduate school and what graduate school has been like, and also describe some of her research, which she will do for us. Those of you who would, if you're interested in what she says, and if you're searching for what to do next, it's not been that long ago that Bethany was searching for what to do next, was in your shoes, was in seminar, and decided to make a change and follow something new. So she's a really great resource to talk to if you would like to talk to her later. Just come up and talk to her. Also, toward the end, we'll have kind of a question-answer time, so be thinking of what you want to ask her. Put her on the spot. So
Please do.
yeah.
Thank you, Dr.
Thank you, Bethany.
Thank you all for having me. I feel like it's not been long at all since I was here, so as Dr. Ahn mentioned, I graduated from King 2 years ago in 2018. And since then, I've been at UT Knoxville in the PhD program. So today I'm just going to tell you about my journey through undergrad, deciding what I wanted to do, changing my mind a bunch of times, which you may relate to that, and then what I've done in the meantime as a PhD in the PhD program. So I was here at King as a student from 2014 to 2018, and when I started college, I thought I would be practical. I thought I would major in biology because I knew I wanted to go to some sort of graduate school, be that medical school or maybe even physical therapy. And pretty much the biology major covered all the prerequisites for that kind of trajectory. And then also minored in Spanish because I knew that there's a lot of Spanish-speaking people in our country. It's one of the fastest growing languages worldwide. So this is what I did. So here on the left, it's just a study abroad trip that I went on to Spain when I was a junior. Just showing all the people there were Spanish majors, Spanish minors like me. And then also I was heavily involved with the Women's STEM program, which hopefully everybody in this room is eligible. And I really enjoyed that as well. So that kind of pushed me into thinking I really, really enjoy biology, really enjoy STEM, and I like interacting with people and showcasing how STEM can make a positive impact in your life. However, I had a problem once I became a junior. In college, I still was not sure where I wanted to go after graduation. So this is kind of the path that I went down and the different career choices I explored as I was a student. So first of all, probably like most people here, you wanted to go to medical school. You might have always dreamed about that. I explored that, I shadowed, I even took the MCAT. And I did okay on it, everything was fine, but I just didn't really feel like that was my true calling. I shadowed in emergency medicine, I shadowed in neurology, and most people would find that pretty interesting, but I just couldn't see myself there. So I moved on from that, then explored a little bit in physical therapy, which my sister Lauren is doing. I shadowed, I took a GRE, and while I enjoyed the clinical aspect, I thought there was also something missing there. So finally, I said, well, I've tried a bunch of things, but I really enjoy teaching. I had realized that about myself as I tutored chemistry here, and maybe many of you were in my chemistry tutoring sessions actually. And I just really enjoyed that. I enjoyed the interaction with students, just learning information, challenging myself to be able to explain it better, helping someone, do better in a subject that a lot of people find hard and challenging. So to further explore that, I then did some summer research at ETSU. I worked in Dr. Stacy Brown's analytical chemistry lab. She came and spoke in a science seminar a few years ago. I'm not sure if any of you all are old enough to remember her. But anyway, I just sent her a cold email one day asking if I could come and work in her lab. And I did that as volunteer work and got some research experience. Kind of last minute. So this is my junior year, right before I was getting to be a senior. And then another thing, just really talked with a lot of the professors here, Dr. Pickard, Dr. Vaughn, about what it's like to be in academia. And I realized at some point, I've been shadowing another profession all along. I've been a student for most of my life. I've been in class, and I really see myself doing this kind of job. So with all these factors together, I decided that research graduate school was for me. Even though I came to King thinking I wanted to be a clinician of some sort. So, having decided that, I had to look at the logistics. Like, can I still apply to research graduate school this year as an upcoming senior? Do I even have the prereqs ready? Well, thankfully, after I looked into it, the prereqs are pretty much the same as what you would have to take in medical school, which I'd already strategically taken care of. So just as you've probably seen before, you need to have biology, all the chemistries, physics, biochemistry, upper level of biology. The only thing I was missing was one semester of calculus. Was my particular program at UT wanted a whole year of calculus. So I actually had a calculus sandwich undergraduate career, because most people take it their first semester when they come, Calc 1. I didn't take Calc 2 until my last semester. So I forgot a lot of calculus in the meantime, but thankfully was able to meet the requirements. Also have the GPA requirement, so this is just the requirement for my school in general. You need a 2.7 out of 4. But then it's really important to have a high GPA in your last year, 3.0 at least. And then, of course, the third thing didn't apply. This is for international students. You have to take a language proficiency test in English. And then, come to find out, now my program has made GRE scores optional. So this is kind of a movement in the sciences in general. Traditionally, the GRE, or the graduate research exam, is the test that you would take if you were going to graduate school. But there is a push in the sciences to remove that as a requirement. So starting with molecular biology is really leading the way, and that's one of the terms in my acronym for my program. Unfortunately, this hasn't yet happened for physics. And as of 2018, about half of the molecular biology programs no longer require the GRE. And then finally, you need some research experience. So hopefully, if you're interested in medical school or research graduate school, you've already gotten some research experience, or are applying for opportunities to get some. But here's just a list of what you can do, and this is something that I was not always aware of. You can actually get some undergraduate research experience here at King. So you heard from two good examples last week. My sister Lauren, Campbell funded her research, and then Marina was funded by the Burke Scholarship. These are all things that you can apply for at King. But then you can also get experience elsewhere, so through like an RU. This is a research experience for undergraduates. And this is also a paid position, and you can do this at another institution. Or if you don't have your act together, you could do what I did and just cold email somebody and volunteer to get some experience. So that actually landed me a little blurb. Through research, I got a little bit of a publication out of what I did with Dr. Brown. So sometimes, even if you're working at the last minute, it can still work out. It's just not recommended. And then, if you are looking for any other RU opportunities, this is one through UT at my school. This is mostly for students that are hard of hearing or deaf. But if you are interested in learning American Sign Language and also in scientific research, this is something you can look into. The deadline is February 14th, so you have a few more days, and I can talk to you after you're interested in this. But my lab is one of the labs that participates in this RU that's funded through the MS lab. Okay. So having decided that yes, I can apply to research graduate school, I've taken the classes I need. This is the procedure that I went through. You have to get all your official transcripts from every place you've ever attended. So not only here at King, but if you've taken something at a community college, you need to send a transcript from it. Pay an application fee. Unfortunately, for every school that you apply to, that's a different fee. UT is pretty cheap. It was about $60, but some places can be over $100 per application. And then you need to write a little research, or a little essay about your research experience, like what you've done so far, what you find interesting, what your career goals are, and then why you want to go to that program in particular. And then finally, you need 3 letters of recommendation. So you can get professors here at King to write you them, or any other mentors. Maybe you have a coach that might be a good resource. And then in particular, if you did research, get a letter from the PI that oversaw your project. So then I applied. I applied to 2 different places. I applied to Indiana University in Bloomington, that's where I'm on my 3rd degree, and then I applied to UT Knoxville in Tennessee. And for the longest time, my inbox looked like this after I had my interviews. There was nothing in it, and I had to wait a while. So that's normal. That might be where you are now or where you will be shortly. Just because you don't hear something for a while doesn't mean it's going to be a bad response when you do hear back. Eventually, I got an acceptance after being on the wait list at University of Tennessee. And this was right around Easter, so it was very fitting. It's good news. So I finally knew what I was going to do after I graduated. So I graduated in April 2018, and I went to the program of biochemistry and cellular and molecular biology. And I just learned this week. The reason that it's such a long name is these used to be separate programs and they combined them. So now we have this really awkward long name for our program. So if you are like me, you might be worried, like, how in the world will I pay for graduate school? Well, that's one of the really nice things about going to research graduate school, is that you get a paid assistantship that will fund you and give you enough money to live on as you're getting your degree. So this can take 2 forms in general. So you could have a graduate teaching assistantship. Most first-year students will have this, where you have to assist a professor with setting up for a lab or teaching a lab course, or maybe you might be grading for a lecture. And it's about 20 hours of work a week. Or if you're a little more advanced in your research and your lab is well funded, then you can be hired as a graduate research assistantship, or a GRA, and basically your boss is paying for you to just do research and you don't have any teaching obligations. So for us at UT, we have about a $20,000 stipend. It's higher elsewhere. It's usually just adjusted with the cost of living, so if the city's more expensive, you will have a higher stipend, but $20,000 is enough to live on for sure. And then on top of that, you actually have your tuition waived. So like normally, if you were a student at UT, you had to pay a certain amount of tuition per semester. That's waived in a PhD program. And then also you get built-in health insurance. Unfortunately, it doesn't include dental and vision at UT, but we're trying to push for that in the graduate senate. So once I got to UT, I had to take a bunch of what looked like very challenging courses. This is just a list of what I had to take. So following the acronym of my program, I had to take a bunch of biochemistry classes, a cell biology class, and then a molecular biology class in the spring. And then every semester I take a graduate research colloquium, which is basically where all the graduate students give research updates about what they're working on, usually just once a year. And then a departmental seminar, which is kind of like what you all do here, where you have sometimes outside speakers, sometimes it will be people from the department giving updates. And then a journal club, where you're discussing scientific papers, learning how to read them. And then probably the most important one of all these is your rotations. So as a first-year student at UT, I had to do 3 rotations, one in the fall and one in the spring, that were 7 weeks long in different labs, in order to get some feel for what the research was like. And ultimately, I learned a big lesson, and I was very thankful to learn this lesson while I was in my first year at UT. And that is that grades are not as important in graduate school as they are in undergrad. If you were like me, you always wanted A's, or at the very worst, A-. But that's not what the emphasis is in graduate school. You need to have at least a 3.0 overall. So getting an A or B in all your classes, but it's not necessarily better to have an A versus a B. It's just a passing standard. So really, in general, if I were to rank the order of importance for all of the responsibilities that you have as a graduate student, It's to maintain your research, make sure you're progressing with that. If you're teaching, you need to be taking care of your classes, your grading, taking care of your students, and then your classroom is after that. So just get the B, B is fine. And that was a lot of stress off my plate. So, any questions so far? Feel free to stop me if I'm going too fast. If you have something you want to chat about, this is informal. Okay. Alright, so at this point, I'm through my first year. I ended up passing all these classes. Some were definitely harder than others. I found the protein chemistry one pretty difficult. Of course, I thought biochemistry was one of the harder classes here at UT as well. At Keene. After I did my rotations my first year, I ended up choosing a lab. That's the DOS lab. So this is my tree DOS is the PI. And we work with fission yeast, or Scutosophomyces pombe, as our model organism, to study how cells grow, how they attain and maintain their cell shape. So this is a basic research lab. I believe if you were in seminar last semester, you heard from Demisha. Her research would be more of an applied research lab where she's addressing like a certain problem. My lab is more just figuring out what the fundamental players are that allow cells to choose a site to grow and how to regulate cell division. And I'll save all that for my talk later, but that's just a distinction. There's a lot of different graduate programs you can go into, some that are more disease model based, others that are more exploratory with basic research. So this is how I chose my lab. First, name all my lab mates. So this is Julie Rich Robertson here. She is a 5th year student in the lab. This is Brian Hersick. He is now PhD. He just graduated in December. My boss, Maitri Das, Dr. Maitri Das, and then Udo Ombiko. She is a 4th year student in the lab. And this is us going to conference in DC this December. And how I chose to join this lab, I took in consideration all the factors I have listed here, and this is something to think about if you are interested in research when you're choosing a lab, is how does that lab make you feel? Does it make you feel welcome? What is the culture and the atmosphere? Do you look forward to going to work every day? You don't want to be in a place where you don't feel like you belong or you're very well supported. So I chose the DOS lab because I feel like I had good support among not only the graduate students, but also from Dr. Das herself. Another thing to consider is the mentorship style of the PI. So some PIs can be more hands-off, and maybe some people like that, and you don't want somebody standing over your shoulder telling you what to do, or maybe you like the security of having someone who's always accessible. And I fall in that latter category. I tend to be a little bit cautious and want to make sure that I'm doing it right, especially the first couple times I do something. So that's something to consider. And thirdly, what is the track record of the lab? Have they graduated with a lot of students? Are mostly the students that graduate getting masters, or are they getting PhDs? That's something that can tell you about the lab. If you have a lot of people who are getting masters, there's usually a reason why. They're not passing the prelims, or maybe it's just not a good work environment. And then also the publication record. You want to make sure that you're joining a productive lab where you're going to be able to get at least a few papers with your name on it, because that's really your currency after you graduate. You want to have something you can showcase as, this is the work I did as a graduate student. It's kind of low on the list, but research interests really aren't the top one priority. You don't want to join a lab just because it has cool science. You want to make sure it's a good place that you feel safe and you like to work. But it should be still something that you find interesting that will keep you engaged for the next 4, 5, 6 years while you're working on a project. And then lastly, something to consider is also the diversity of the lab. Like if you are a woman, do you want to join a lab where it's all males, and you don't want to be the oddball. You want to make sure that you're going to feel comfortable working there. All right. Any questions? So this is kind of a hypothetical day for me as a graduate student on the right. So this would be if I was teaching. I don't teach every semester. I'm actually not teaching right now. But usually when I do, I ask for the morning time. That way I can just go, to class, teach for 2 hours. I usually teach a wet lab. I have been teaching non-major biology, so I get to interact with a lot of people who maybe are not that much about science, but it's kind of fun because they can learn that, oh, this is actually pretty cool, or I've never thought about this. So I teach usually in the morning, and then I might have a little bit of downtime before class. I put my journal club on there, and that's just once a week. Then I usually take lunch from about 1 to 2. Then I might do some imaging on the microscope, because that's what we do as cell biologists. That's a lot of what I do. And then could have seminar, followed by just time for dinner, and I need to go home and check on my dog Sadie. And then I might have some obligations in the evening where I have meetings, maybe for the Graduate Student Senate, which I'm a part of, or I can do some tutoring, or maybe even some volleyball. And then I usually, before I close the day, go back to doing some more reading, making sure I'm keeping up with the literature in my field, or doing some data analysis. So all in all, on the average week, and I spend about 20 hours teaching. Usually it's not quite that much, more about 15. Wet lab and imaging, all the data analysis, I might spend over 30 hours a week working on that. If I have a lot of classes, it could be up to 8 hours a week. Meetings, including lab meetings or individual meetings with your boss or any other program that you might be involved with, could be about 4 hours a week. And then if you're keeping up with your field, you should be reading about 8 hours a week, so cover about an hour to 2 hours a day. I don't always hit that, but that's what you should aim for. So if you're keeping track, that's about 70 hours of work. So it's busy. I probably don't hit 70 hours every week, but there are definitely some weeks that I'm there with everything that I'm doing, with my teaching obligations, research, and classwork. It takes a lot of time. It's very fun. I do have some free time too, though. I play volleyball a lot. We have a group in my department, and then we also invite people from micro to play with us. In the summertime, we play sand volleyball, and then in winter, we usually play just in the indoor student center. Thankfully, around Knoxville, there's a lot of hiking and outdoor activities. So I will go paddleboarding. There's my dog Sadie on the paddleboard. And then this is at the Norris stand. It's kind of like a South Wilson dam. I went there a couple weeks ago. So just because you're working a lot doesn't mean that there isn't time for fun things as well. All right. So this is really the natural breaking point of the middle of my talk, because now I'm going to start talking about my research a little bit. So if you all have questions about graduate school yet, or you want to hold them, it's up to you all how you want to conduct this. Okay, questions? Okay, well, we can have a panel discussion at the end where you can ask me all your questions. So transitioning now into more of my research, I'll just read the title for now and then explain as I go along. So as you know, I work with S-Palmy. That's my model organism. They are a type of fission yeast, or type of yeast that's called fission yeast. It's a cousin of budding yeast. They're not too, they're pretty well related, but not super close related. And I'm working right now on characterizing the function of a gene called PPS1, which is a phosphatidylserine synthase. So phosphatidylserine is a lipid that comprises the membrane of these organisms and all the way up to other eukaryotic organisms like humans. So this is a lipid that is important for cell signaling and, as I will show you, cell growth and maintenance of cell dimensions. So first of all, in general, my lab studies cell polarity and cell growth. So what is cell polarity? Well, it's how cells maintain and attain their proper shape. So this is important for all organisms. So if you have cells in a tissue, like in a muscle tissue, or in the lining of your GI tract, those cells need to have a certain shape in order to stay anchored to their neighbors. And if they start growing or dividing in ways that they shouldn't, this can lead to cancer. So this is, the basic research in the bottom, level of what can be applied to cancer biology. Cell polarity also regulates how cells proliferate and how they differentiate during development. If you've taken developmental biology with Dr. Ong, you probably learned about this. And it just helps cells know who their neighbors are and where they are supposed to be located. And this is all regulated by the actin cytoskeleton, which is it functions kind of like an interstate system in the cell where you can take cell components to a certain site, deliver those materials so that the cells have what they need to expand the membrane and grow the cell in a certain area. So since we work with S-POMI, let me tell you about how it grows in general. So it is a rod-shaped organism, like you can see here, so it has 2 cell ends. And it goes through this type of cell cycle and cell division. So let's start over here, where this is a dividing cell. So these are actually cell-walled organisms. And they have to produce not only the membrane, but an outer cell wall that protects them. So here in green, you're seeing the synthesis of what will be the new cell wall after these 2 daughter cells divide through cytokinesis. After that happens, there's what's called an old end and a new end. So if you can kind of match the green here, so here there's the new end. After this cell divides, let's just say for this one. After this cell divides, you'll have what's the new end and the old end. So the old end is what existed in the previous generation. The new end is synthesized by the division of these 2 cells. And after the division, the old end is going to be the end that grows first in what's called monopolar growth. And as far as we know, and it's always seen in all our various mutants, fission yeast always grow in the old end first. We're still working on figuring out the mechanism of why that's the case, but this is what we know so far. After the cells then grow to get a little bit longer, they then transition to bipolar growth, where now both ends are growing at the same time. So all of this is interphase, as cells are growing until it's time to divide again. So here they reach their correct length, which is about 14 microns. And now they're going to start constricting their actin myosin ring, which is very similar to what we have in animal cells. They're going to pinch the membrane in and then start to divide, and then they're going to build in their cell wall in behind in the septum to then divide. So in general, fission yeast grow like this. First at the old end, then transitioning to bipolar growth, and then dividing once they reach the right length. And this is all regulated by a molecule called CDC42. So this is a Rhode family GTPase. It's a subfamily of the RAS category of signaling proteins, which have implications for cancer biology. If RAS is unregulated, then you can have unregulated cell growth. And this molecule coordinates the delivery of all of these materials to the membrane in order to expand it and help the cell grow. So, as I was telling before, you have these actin cables, which are like interstate systems in the cell, where there are cargo carriers like myosins that walk on them and actually deliver these components to the cell tips where fission yeast grow and help them to establish this zone of growth, which is basically cell polarity. This is all regulated by a molecular mechanism that occurs at the cell membrane. So, like other Rho GTPases, CDC42 is activated by guanine exchange factors, and they activate it, telling it to be on, and then it has deactivators called gaps that are listed here that turn it off. And this all happens through exchange of GDP and GTP. So when CDC42 is bound to GDP, that's going to power the molecule to create the delivery of all of these components. And then when it has GDP, then it's going to be turned off, so there's no delivery. Does that make sense? Okay, so basically CDC42 is a molecular switch that's either on or off, depending if it's bound to GDP, active, or GDP, inactive. So let me walk you through what we currently know about CDC42 regulation. From my lab, And others, we've learned that there are 2 GEFs for CDC42, that 2 activators. They need 2 because there are 2 zones of growth, basically, in fission yeast. So SCD1 is the main GEF. It's the one that is most similar to the main GEF embedding yeast, so it's one of the better studied ones. And then there's also another GEF, GEF1, that promotes bipolar growth. So if you were to delete GEF1, then you would not have the transition to both cell tips growing, you would just see one tip growing. And then we have the gaps, which restrict CDC42 to just the cell tips. And I failed to mention before, they're located along the cell sides, where CDC42 cannot be active here. This helps the cell to grow just at the cell tips. So CDC42 is inactive and only active here in green. And all of these proteins, including CDC42 itself, have membrane binding domains. I've shown them here in red, so what you're looking at here are just a cartoon illustration of the different domains or the different regions of these proteins and what they do. So here are the GEFs. They have like their catalytic GEF domains that activate CDC42. RGA4 and RGA6 are the deactivators, so they have their GAP domains that deactivate CDC42. But they also have all these domains shown in red, which are known from the literature to bind to membrane. So what my research question is asking is, if we know how CDC42 is turned on and turned off, how can we figure out how it gets to the membrane? In order to say this is a site that the cell wants to grow, how are we going to recruit those players to that site in order to build the membrane there? So this is where my project comes in. There has been literature, it is known in the literature that anionic or negatively charged lipids, so this is like the components of the membrane that build the cell, are negatively charged, and they can actually recruit positively charged proteins like CDC42. So it has a CAX domain, so this is just 4 amino acids, on its C-terminal end that is actually attracted to negative lipids like phosphatidylserine. And this is not trivial because in yeast, the membrane is composed of about 20% of PS on the inner leaflet. So if you can imagine, this is the inside of the cell. The lipids here are negatively charged, and you're attracting a positively charged protein to that site, and then it's going to become active and then start signaling for the delivery of all these cell building materials in order to say this is where we want to grow. We're going to start expanding the membrane here to establish proper cell shape. But this is not enough just for CDC42 itself to interact with the membrane. You need to have the localization of its GEFs or its activators, because you could have random deactivation through the gaps as well. So you want to keep CDC42